So imagine this, like a world-altering plague, a ghost from history, that for 1,500 years really haunted historians.
Like, it reshaped entire empires,
Yeah.
wiped out huge numbers of people.
And then the actual cause, it just remained the scientific cold case. Well, until now.
Exactly.
Today, we're not just, opening a history book. We're actually looking inside ancient teeth from a mass grave in Jordan.
And finding definitive genetic proof. Yersinia pestis. Right there,
Yeah.
at the heart of the Plague of Justinian.
Yeah, and this isn't just about saying, aha, we knew it.
Right.
It's actually rewriting the whole story of how plague emerges, how it evolves, how it sticks around.
And it offers some really vital lessons, maybe even chilling ones, for us today.
Absolutely. So Plague of Justinian, AD 541 to 750, often called the first pandemic, its impact, huge, on the Byzantine Empire, on Western civilization.
But for centuries, the specific bug behind it all was debated. Hotly debated.
So our mission in this deep dive is to unpack this huge finding from Jerash, connect it to the surprising bigger picture of plague history.
And figure out what these ancient germs can teach us about public health now.
Okay, let's get into this ancient mystery.
So for centuries, you have these historical texts describing the Plague of Justinian as just massive calamity.
And historians suspected Yersinia pestis, right? The plague bacterium we know.
They did, yeah. The circumstantial evidence was really strong. Descriptions of fast-spreading fever, the buboes, incredibly high death rates, symptoms that sound a lot like later plague outbreaks. But despite all that, actual biological proof, especially from the Eastern Mediterranean, where the historical accounts said it all started, it was missing.
The missing link.
Exactly, a critical missing link. Like knowing a crime happened but having no fingerprints from the scene.
And that's where this new research comes in, groundbreaking stuff.
Absolutely landmark. You've got this interdisciplinary team, University of South Florida, Florida Atlantic University, international partners too.
And what do they find?
They identified Yersinia pestis DNA, actual genetic material, inside 1,500-year-old human teeth.
Wow. From where exactly?
From a mass grave site in the ancient city of Jerash in Jordan.
So this isn't just another clue. This is like the smoking gun.
It really is. It's the centerpiece. It firmly puts Jerash on the map as, ground zero for genetically confirmed plague during that first pandemic.
Hadn't they found Y. pestis DNA from that era before, though?
They had, yes, but that's the key difference here. Those previous finds were thousands of miles away. Think small villages in Western Europe, scattered finds.
Okay, so important, but maybe not the origin point.
Right. They didn't tell us much about where it started or how it initially spread. This Jerash discovery is huge because of where it is, geographically.
How so?
It's only about 200 miles from Pelusium in Egypt. And Pelusium is exactly where the ancient texts first described the plague appearing and just exploding.
Okay. So it connects the dots?
Perfectly. It connects the historical accounts with the hard genetic evidence, right there near the suspected origin. We're not just confirming the bacteria, we're placing it geographically at the start of the story.
It's just mind-boggling to think about getting DNA from teeth that old. How on earth did they manage that? What was the process?
It really shows how far ancient DNA technology has come. It's amazing stuff. The team, you had archaeologists, geneticists, all working together.
A real team effort.
Definitely. They use these really targeted techniques, specifically designed to hunt for tiny, fragmented bits of ancient DNA unique to Yersinia pestis within those teeth.
But just DNA?
No, and that's another clever part. They also did what's called preliminary proteomic screening.
Proteomics, so proteins.
Exactly. Think of DNA as the blueprint, right?
Yeah.
Proteins are more like the tiny machines the bacteria build based on that blueprint. And some proteins are incredibly tough, really durable.
So they can last even longer than DNA sometimes.
They can, yes. Even after 1,500 years, these protein signatures can hang around. They act like these incredibly resilient fingerprints that help confirm the pathogen was there.
So it's like a double confirmation.
Precisely. This careful, multi-pronged approach, genetics plus proteomics, let them not only find the material, but sequence it, rebuild the Y. pestis genome from these ancient samples. It's just irrefutable proof.
Like finding bits of an old manuscript and then confirming the ink in the parchment, too.
That's a great analogy, yeah.
Yeah.
Very robust.
Okay, so Jerash. It wasn't some small village. It was a major Roman city, right?
Right.
Thriving. But this discovery wasn't in a nice, neat tomb, was it? It was a mass grave.
That's right. A mass grave.
Tell us more about that. What does finding plague victims in a mass grave tell us about how bad things got? What did that site look like?
It's incredibly stark. A really chilling picture of societal breakdown. These samples came from burial chambers underneath the old Roman hippodrome.
The Shippodrome, like where they had chariot races.
Exactly. Imagine this grand stadium built for entertainment, for public spectacle, probably mid-2nd century AD. And then, suddenly, it's repurposed, turned into a huge makeshift cemetery.
Wow, how many people?
They found about 150 adults and around 80 subadults, so babies, infants, children, packed into these chambers.
Just overwhelming numbers.
Completely.
Yeah.
The sheer number of bodies, the fact they had to use a major public venue like the Hippodrome as an emergency graveyard.
Yeah.
It happened right in the mid-6th to early 7th century AD.
Which lines up perfectly with the historical accounts of the plague hitting hard.
Perfectly. It screams of a situation where the number of dead just completely overwhelmed the living. Normal burial practices, which were deeply important culturally, just went out the window. Total crisis mode.
You read about this stuff, but picturing a hippodrome filled with bodies.
It mirrors exactly what chroniclers described in Constantinople and other big cities at the time, streets overflowing, nobody left to bury the dead. Jerash gives us this tangible, horrifying glimpse into that reality.
It really grounds it, doesn't it? Moves it from abstract history to human suffering. Those teeth belong to real people caught in this nightmare.
Absolutely. It connects us directly to them.
And the genetic evidence itself from those specific Jerash victims. What did that reveal about the outbreak itself? Was it one specific type of plague?
It told us something really significant about the speed and the nature of that particular outbreak.
Yeah.
The genomic analysis showed that the victims there, they carried almost identical strains of Y. pestis.
Identical. What does that mean?
That genetic uniformity, that lack of variation, it strongly suggests a single strain just ripped through the population very, very quickly. A rapid, devastating, localized explosion.
Which again matches the historical descriptions of sudden widespread death.
Exactly. And importantly, this Jerash strain, it had the genetic toolkit for causing serious trouble. Things like the IMPIT, PLA, and F1 capsule genes.
Okay, technical terms. What do those genes actually do?
Think of them as the pathogen's weapons or special tools. They produce proteins that help it hide from the immune system, multiply like crazy inside the body, and spread efficiently, often using fleas as vectors to new hosts.
So finding those specific genes confirms it wasn't just any old bacterium. It was a highly dangerous, well-adapted strain.
Precisely. It had the necessary virulence factors to be incredibly pathogenic.
And seeing this in Jerash, an urban center, what does that tell us?
It gives us this rare, direct look at how an ancient society just buckled under a public health disaster. And it really highlights how cities, these hubs of trade, travel, dense populations, were just incredibly vulnerable.
Because everyone's so connected.
Right. The very things that made late Roman cities vibrant, the connectivity, the movement of people and goods, ironically created perfect highways for a pathogen like this to spread rapidly. Jerash became a tragic example of that urban vulnerability.
So, okay, this Jerash find is huge for confirming the Justinian plague. But you mentioned it also changes our understanding of plague overall. How so? Does it connect to later plagues like the Black Death?
That's where things get really interesting and maybe even counterintuitive. The assumption was often, a single line. Justinian's plague evolves into the Black Death strain, which evolves into modern strains.
Right, like one continuous story.
But this research, especially a companion study looking at the broader genomic picture, turns that idea on its head.
Oh.
It shows that later plague pandemics, the Black Death in the 14th century, even the strains circulating today, did not simply descend from one single ancient source, like the Justinian strain.
Wait, really? So they weren't directly related?
It seems not, in a direct lineage way. Instead, the evidence suggests they emerged independently, repeatedly, popping up again and again from these long-term animal reservoirs.
Wow. So plague isn't one monster. It's like a recurring nightmare that keeps finding new ways out of the shadows.
That's a good way to put it. It challenges this whole idea of a single linear evolution for plague pandemics. It's much more complex, this pattern of repeat emergence from hidden animal populations, likely driven by environmental factors we're still trying to fully understand.
That is a massive shift, especially when you think about other recent pandemics. So let's make this concrete. How is Yersinia pestis' evolution so different from, say, SARS-CoV-2, the virus behind COVID-19? We all tracked the variants with COVID, right? Alpha, Delta, Omicron seemed like a clear progression.
Exactly. There's a perfect contrast. SARS-CoV-2, as far as we know, came from likely a single spillover event. And then it evolved mainly through continuous person-to-person spread.
And you could track it.
Right. It showed a pretty clear molecular clock. Its genetic changes piled up at a relatively steady, predictable rate. That's what allowed scientists to trace its family tree, see how Delta evolved from earlier strains, how Omicron branched off. It was clock-like, predictable.
Okay, so how is Y. pestis different then?
Y. pestis doesn't follow that simple clock. We talk about it having a cryptic clock.
Cryptic clock. Meaning hidden.
Kind of. It means it lacks a single global molecular clock that ticks along steadily. Its evolution is nonlinear. It seems to have long periods where its evolution slows right down, almost goes dormant, when it's just hanging out in its animal or environmental reservoirs.
So it hits pause when it's in animals?
Something like that. It's not evolving rapidly in that state. But then when it jumps into humans and starts causing a major epidemic, like during the first pandemic or the second pandemic.
It hits fast forward.
Exactly.
Yeah.
That's when we see the clock-like genetic changes appearing, the rapid evolution happening, driven by intense human-to-human transmission. Then it might retreat back into reservoirs and go quiet again for potentially centuries.
So it can just wait.
Yeah.
And then reemerge, maybe looking genetically similar to how it did centuries before it pops up again.
That seems to be part of the picture, yes.
Oh.
Which makes tracking its deep history much trickier than something like SARS-CoV-2.
That's fascinating. And you mentioned reservoirs. Does the type of host it prefers also play into this? You said something about ancient strains versus modern ones.
Yes. That's another really crucial difference they've found. When you look at the ancient strains from prehistory, from Justinian's time, like in Jerash, and from the Black Death, where do we find them?
Only in the human remains.
Well, exclusively.
Yeah.
That strongly suggests that during those massive pandemics, the bacterium was primarily adapted to and spreading fiercely within human populations. It was acting like a human-centric disease during those outbreaks.
Okay. And modern plague?
Modern Y. pestis lineages, the ones causing cases today, we find them all over the place.
Yeah.
And a whole range of hosts, different rodents, their fleas, sometimes even in the soil or environment.
So it shifted its strategy.
It seems to have shifted its ecological niche, yes. From being mainly a human pathogen during those historical crisis periods to being a more general zoonotic disease today, cycling mainly in animal populations with occasional spillovers to humans.
So putting this all together, these pandemics aren't just one-off historical disasters.
Not at all. They look more like repeating biological events, driven by this complex dance between, how humans live, our density, our travel, environmental factors affecting those animal reservoirs, and the incredible adaptability of the pathogen itself.
A pattern repeating across history.
The deep recurring pattern, yeah. Reminding us these cycles are embedded in the natural world.
It's so easy to think of plague as, medieval history, something safely locked away in the past. But it's really not, is it?
Absolutely not.
Yeah.
Yersinia pestis is still out there. It's an active threat today.
And we see reminders. You mentioned recent cases even here in the U.S. Someone died in Arizona in July, another case in California just last week.
Exactly. Those are stark reminders that this ancient pathogen is still circulating globally in animal reservoirs. It hasn't gone away.
And unlike some diseases, we can't just eliminate it, can we?
No. That's a critical point. Like COVID-19, in many ways, plague is a pathogen we probably can't fully eradicate. It's too embedded in those animal reservoirs. It keeps evolving, adapting.
An uncookable pathogen, you called it.
Yeah, maybe that's one way to think of it. We can manage outbreaks, use antibiotics to control rodent populations, but getting rid of it entirely from the planet, probably not feasible because it's fundamentally a zoonotic disease with deep roots in wildlife.
So this forces us back to the lessons. What can Jerash, what can this long history of plague teach us about preparing for the next thing, whatever pathogen emerges?
I think one huge lesson is the enduring power of connectivity. Trade routes, dense cities, global travel. These things fueled prosperity in the past, and they do now. But they also create the pathways for pandemics.
The double-edged sword of interconnectedness.
Precisely. The Jerash research just hammers home that link between connectivity and risk. The very structure of ancient urbanism made places like Jerash vulnerable, just like our globalized world makes us vulnerable today, maybe even more so.
So studying these ancient outbreaks isn't just history, it's actually vital for modern public health strategy.
It really is. It's becoming a crucial part of understanding disease dynamics, and the research is pushing forward.
How so? What's next?
Well, for example, teams are now studying other historical mass graves, like those from the Black Death era on Lazaretto Vecchio. That's one of the quarantine islands near Venice.
Looking at how they tried to manage it back then.
Exactly. Studying those sites can tell us so much more about early public health responses, how they interacted with the pathogen's evolution, how cities tried to cope, the cultural memory of these events. These ancient pathogens, especially when we unlock their genetic secrets, are sending us vital messages across time.
Messages about our own vulnerabilities.
Absolutely. About what it means to live in a world where pathogens constantly adapt and challenge us.
Okay, so this deep dive has really shifted my perspective. We've gone from a historical mystery to definitive genetic proof from Jerash, confirming Yersinia pestis in the Plague of Justinian.
Right, finally closing that 1,500-year-old gap.
And we've learned about this totally different evolutionary path for plague, this nonlinear cryptic clock, the deep roots in animal reservoirs, the repeat emergences.
And how different that is from something like SARS-CoV-2.
Yeah. So here's the thought I'm left with. If plague, this ancient disease, can just keep re-emerging from hidden reservoirs, and if our interconnected cities basically amplify its spread, what other ancient pathogens might still be out there?
Silently waiting or quietly evolving?
Exactly. In reservoirs we don't even know about. And seeing these deep repeating patterns of disease across history, how prepared are we really for their inevitable return in our hyper-connected world?
That is the crucial question, isn't it?
It definitely connects us right back to those people in Jerash, doesn't it? A shared vulnerability across millennia. A sobering thought to end on.